Good morning. I welcome you on our SOPHIA studio, and it is a pleasure to have you with us for an exchange of views and on the issues going on this Deep Sea Symposium. I understand that both of you have shared the working session, the panel on in advances in taxonomy and phylogeny. It's something of they are heavy words, don't you think so? Most people, when they talk about taxonomy and phylogeny, they don't know what's going on. No. So, could you please make us a very small briefing on the outcome of this session? I would, if you allow me, to start with Dr. Gordon Paterson, that comes from the UK and Natural History Museum. So, what is going on, please?
Yes, I'm Gordon Paterson. I'm from the Natural History Museum in London. Taxonomy is the foundation block of all of biological science. It's the discipline about identifying and naming species. And the session that we had today was explaining what the new techniques and the new ways we're looking at identifying the fantastic diversity and species richness that you find in the deep ocean. So we had a very interesting range of speakers from lots of different countries who are applying a whole range of very exciting new techniques using modern molecular methods, using more traditional techniques but in a more intensive way, more considered way, to come up with new information and new insights into the biodiversity of the planet. And the deep sea is a fantastic place because we know so little about it. So nearly everything we get and sample and encounter is new. But what's more important as well is to work out how these organisms are related to one another and how the system evolved, because that gives us insights to the kinds of pressures that we are now putting on our planet and how these animals are going to respond. So today was a very interesting session. Encouragingly, we had lots of young people who gave presentations, parts of their PhDs. So it's very good to see that these people are taking on board this very old discipline, because taxonomy is as old as mankind. We've been classifying and identifying things since the beginning. But what these new people coming to this old discipline are doing is they're bringing new methodologies, new approaches, new insights. And that's very exciting because you know then that we're going to advance our knowledge, and it's going to reveal a lot more interesting and exciting results from the studies on the deep ocean.
Oh. And I think there is a very important issue on this, because those young people bring new life to an old part of the biological science. Exactly. So it's an hope for the future. It's the way forward. Yes. And also, I retained from your excellent explanation that the deep sea is not something dark and devoid of life, on contrast. No. Yeah? No. And it is something new to our minds.
It's one of the most exciting places to work on the planet, because the diversity is as high as you get in tropical rainforests in some areas. And it's all new. Yeah. And it's also the biggest environment on our planet. And so we tend to think that because we can't, as human beings, experience it in the way that we experience the world around us, we tend to ignore it. But when you make the effort, when we put the technology to go and explore with our new submersibles and our remotely operated vehicles, the wonders that we see under there are amazing. The new forms of life, from small worms and bacteria right up to big fish and squid that are 20 meters long, that's only just been discovered in the last 20 years. It's absolutely phenomenal.
And they have been always there.
Yes, exactly. Yes. We just never found them before.
We just live parallel lives without knowing each other. It's amazing. So, and the other thing that is really amazing to the people looking for this issue from the very first time is to find Portuguese people, young Portuguese people involved in these international teams and going ahead in an excellent way. So we are not shy of going to work in world-class teams. And it is also something that usually is not perceived by the layman in the street. Say, those are the Americans with satellites and so. But no, we have also those guys somewhere with us in our universities. And so I would like to ask Dr. Ascensão Ravara to tell us a bit. You are a researcher here at CESAM in the University of Aveiro, and I see you have a very an excellent track record on celebrated projects like Hermione and Hermes, and so you have been researching also marine benthic ecology and biodiversity with a special interest in polychaetes, and something that interests me also. What are those cryptic species? Not all species are the same. Please tell us.
Yes, cryptic species are species that are morphologically very similar. That sometimes you look at specimens that you cannot tell that they are different. You cannot separate them. But then when you go deeper, when you use other techniques, molecular techniques, for instance, then you realize they are actually different species. So those are cryptic, are what we call cryptic species. Maybe there are some morphological differences, but we cannot see at the first time. We have to look more careful, maybe, or apparently, or use other techniques like SEM, like scanning electronic microscopy, and things like that to go further to go to detail.
If you look into a crowd from the human point of view, if you look into a crowd, everybody are similar. Then you realize they are not. Each one is an individual.
So there's been lots of, actually on the last decades, on last years, we found lots of new species from that cryptic, that we believe there was only one species. We have been, with the new technology, we have been finding lots of new species.
It's an amazing new age of discovery.
Yes. So we keep. So that's where the museums get in also, because many times you have to go back to the old material and look again and read, describe it, and
and also making sure that materials that you are collecting at huge cost, physical and financial, and so will be there and be able to be studied in the future.
You cannot go always on the no, I have to go there on sea and collect some more specimens. It's not exactly like that. So it's really important to have some place where you can keep the material in good conditions and all the metadata associated with it, so you can further study it.
Okay, and I would like to ask one question. Is that easy to keep those collections, or people just in the financing sector and political area just think a mission is going there, making a report, putting a video somewhere, and then that's all? That work of keeping things and long-term investment, is it easy to make it perceptible to decision makers on economical scale, they understand?
Not always.
But I think the issue to get across is that this is an archive of life on the planet. And the interesting thing is that when we look at what's happening now, often we can compare with how much things have changed by going back into the collections, by getting the information from the collections. That gives us the context of the change that we're facing, whether it's happening quickly or whether it's happening slowly, whether it's happened before. If you go back far enough, you can look at fossil collections, and using those collections, you can see how the planet has changed in the past. And then you can compare with how the planet is changing now and how biodiversity is changing. Well, if we didn't have collections, if we didn't have the information about collections, that would be so much more difficult.
Yeah.
And so the collections themselves are an incredible archive. For biodiversity, collections are the equivalent of the Large Hadron Collider facility. People come and use our collections daily. They study the material we have, they look at the data, they compare it with theirs, and the basis of taxonomy is this comparison that is made, either using new material that we might make molecular collections with, or looking back at the traditional and marrying the two. This is fundamental for science.
Yes.
If you don't have this baseline, you don't have this identification, then you are not able to say things. And new diseases and new invasives occur all the time. The way we identify those is because we can compare them to the museum collections. So something that may be economically really very important, maybe a pest to crop, or it may be something coming into fisheries that may have an adverse effect, an economic adverse effect on ordinary people. How do you identify that that's happening? You do that because you know what you have a fauna. Yes, and you have the baseline, and that's where the collections come from.
Okay. Scientists realize they are very important, but we must put that message to the society, in order to show those people that it's not a dump of material, of biology.
Well, we also try to do that by having visitors and also from the schools. Yes, the youngsters are our future, right? So we try very much to tell them how important it is, and to show them how nice it could be also to see. So it also fits teaching purposes and
I fully agree with both points, and I would like to thank you once again for your willingness and availability of expertise for the proposals of our project SOPHIA. Thank you very much.
Thank you for inviting us. Thank you.